My name is Abdullah Sheikh and I am here with Darpan Khanna, and today we will be discussing our research on exploring protein structure dynamics using graph-based attention and Voronoi tessellation saliency mapping. The motivation of our research is that protein structure prediction and analysis are vital in structural biology and bioinformatics. They offer insights into protein mechanisms, bondings, and interactions. This understanding is crucial for applications like biotechnology and drug discovery, aiding in the development of targeted treatments and advancing biological research. Despite breakthroughs like AlphaFold in predicting single-chain structures, deep learning techniques have not matched the accuracy of classical methods for multimeric structures in Cas13 datasets. Our work aims to bridge this gap and improve the precision of protein structure prediction. Objectives of our research are enhancing the understanding of protein-protein interaction interfaces, applying Voronoi tessellation and graph attention networks for detailed analysis, and finally investigating the influence of amino acids and structural properties on the protein-protein interaction stability. By validating our computational methods, we aim to improve prediction of protein stability and interaction dynamics, contributing to more accurate and reliable protein engineering. Structure of protein interfaces, we use a concept called Voronoi diagrams. First, let's define what Voronoi tessellation is. Voronoi tessellation divides a plane into regions based on the nearest points, which we call seeds. Each region contains all the points that are closest to its seed. In the image on the slide, you can see blue dots representing the seeds. The lines around each seeds form regions, and every point within a region is closest to its corresponding seed. Now, let's talk about the application of Voronoi tessellation in our 3D protein structures. This method helps model the spatial distribution of atoms within the protein. By doing so, it aids in analyzing the protein structures and identifying how the atoms interact with each other. The second most important element of our research is the graph attention network model. We use graph attention network model to accurately predict the stability, or in our case, the CAD energy score, which implies stability of the protein models, and train an attention network model to predict and accurately classify the stability of the various protein that goes into it. So our graph attention model consists of 4 different layers, and it has 3 different activation layers called LU activations, and the graph layers are convolutional graph attention network models. The model takes input of node features and edge attributes to calculate the structural stabilities and output them in the form of CAD energy scores. This model was trained on 14,000 different graph models and validated and tested on another 2,800 models. Right. To consolidate the process, we begin with raw protein data and convert it into Voronoi tessellation using the methodologies previously explained. We then employ a rule set to transform these tessellations into a 2D multigraph structure. These graph structures are inputted into a graph attention network where the model utilizes neural networks and attention mechanisms to predict protein stability. Following this, we apply saliency mapping, a technique that highlights where the model is focusing within the protein data. And finally, we use this saliency mapping to correlate back to the raw data and conduct meaningful analysis.
Once we are done with the three Voronoi tessellation, we get the graph representation of our protein data as you can see from the first image. We can get a multi-graph dataset with which reveals saliency analysis factors in protein stability. We get insights into the hydrophobic interactions which are very crucial for protein-protein foldings. As you can see from the first picture, it is much more complex and more detailed in giving us information as compared to the older and previous method as shown in the second image. This utilized this is utilized by graphene protein structures. These protein structures are very much fundamental and it does not give enough information. Hence, the Voronoi tessellation generated graphs are better. The first image shows the summed saliency importance of amino acids in protein-protein interactions according to a graph attention network, highlighting the crucial role of hydrophobic interactions in protein folding. Hydrophobic amino acids like isoleucine and leucine enhance stability significantly more than glycine, with 81.48% and 80.77 higher contributions respectively. This emphasizes the importance of a stable hydrophobic core for proper protein folding. The second image displays the frequency distribution of amino acids based on saliency activation, revealing that amino acids like leucine significantly contribute to the model's saliency. The frequent occurrence of such amino acids correlates with increased stability, influencing the model's learning process and predictions on protein stability. Thus, the natural abundance of these amino acids is crucial for interpreting the model's outcome. Further analysis on the saliency importance of node degrees also revealed that nodes with degrees 5 and 6 show higher saliency levels. This indicates that optimal interaction degrees enhance stabilization while avoiding steric hindrance. The heterogeneous distribution of saliency highlights that not all degrees contribute equally to the model's predictions, but suggesting that specific interaction patterns are more critical for maintaining protein stability. Furthermore, in the graph comparing Voronoi energy, which basically is a mathematical relationship between the size and volume of the atoms in the protein, from the image we observed a direct correlation between the compactness of protein structures within Voronoi tessellation and their predictive importance in the model. Regions characterized by lower Voronoi energy correlate with higher structural stability. This indicates that more stable and compact structures have lower Voronoi energy, underscoring the importance of geometric compactness for protein stability. In our research, we used visual saliency within the graph attention framework to analyze protein stability and interactions, identifying key factors like amino acid properties, node degree, and Voronoi energy as critical for stability and structural integrity. Future work will apply these insights to dynamic protein modeling to predict real time effects of mutations on stability, enhancing drug development. We also plan to extend our graph based methods to uncover novel patterns in biomolecular data, broadening the biotechnological and medical applications. Thank you.